I'm Zoe Benedict, and I focused on carbon nanotubes for cell scaffolding. Tissue engineering is reliant on highly porous scaffolds to generate tissues in a template formation that mimics the ECM microenvironment when combined with growth factors. Scaffolds allow departure from 2D monolayers by allowing 3D porous constructs that cells can seed into and throughout. The requirements for a scaffold are that they are biocompatible, biodegradable if going into the body, have good mechanical structure to avoid collapsing, and have a porous scaffold architecture to allow proliferation throughout, as well as good feasible manufacturability. These scaffolds can be used for in vitro tissue generation for implant, in vivo prosthesis and implants inside of the body, as well as in vitro test setups to study cell interactions for treatments. CNTs contribute to nanofibrosity that helps to mimic the ECM and the microenvironment that the desired tissue would normally be inside of. The structure helps to supply growth factors, which in turn better functionalizes tissues. CNTs are formed from graphene nanolayers that are rolled into hollow tubes with nano-sized diameters. There are two types. Single-walled carbon nanotubes are single-layered tubes with very small diameters ranging between 0.5 and 1.5 nanometers. Meanwhile, multi-walled cellular carbon nanotubes are layered concentric tubes and tend to have larger diameters in general. CNTs are desirable for scaffolding due to their improved mechanical properties in small concentrations, their ability to be sterilized, their simple synthesis and functionalization, good elasticity characteristics, good electrical conductivity, which is useful in cardiac and neuronal tissue stimulation, as well as their modifiable wettability, which allows them to have good hydrophilicity for cell adhesion. CNT has been found to be a good scaffolding material to differentiate and proliferate bone, cardiac, and muscular cell tissues. It has also been explored for neuronal tissues due to its electrical charging ability, but prostheses are limited by long-term cytotoxicity. Typically, CNTs are incorporated in composite layers or as additives to polymers because of the good properties that can be achieved with small dispersed concentrations. For example, it makes for a good reinforced material such as CNT reinforced collagen for bone tissue generation, and it can also be treated for adjusting the hydrophilicity. However, due to its nanosizes, it is hard to disperse because they like to agglomerate, and it has bad interfacial adhesion to composites. The alignment of CNT matters because the vertical rods tend to interpenetrate the cells, while random orientation facilitates a good growth bed. The electrical conductance can also be adjusted for experiments of electrical stimulation on growth due to percolation theory. The problems with CNTs lies in their acute cytotoxicity. CNTs and scaffolds are typically loosely aggregated and can easily be released into the body during disintegration during inflammatory reactions. Also, due to the smaller diameter of single-walled CNTs than multi-walled CNTs, they are more readily able to impair phagocytosis. However, both types are capable of apoptosis, a type of programmed cell death, as well as fibrogenesis. In a flow cytometry study of single and multi-walled CNTs, apoptosis on MC4L2 cells in mice, similar results were found between them, with each being capable of up to 65% of cell apoptosis after 72 hours. One solution to this problem that has been explored is to use thin covalently bonded coatings to provide better CNT adhesion. However, until the cytotoxicity issue can be improved, it has mostly been utilized for in vitro studies of cell interactions, such as growing tumors for drug testing. CNT scaffolds are good options for experiments in vitro cell growth, and their customizability makes them ideal for being able to grow many types of tissues. However, due to their easy cytotoxicity and fibrogenicity when they are disintegrated, there's a lot of room for improvement before they are used for in vivo scaffolds.